So Chris is the head of the Department of Engineering at King's College London, and he's professor of computational material science. So a short summary of his bio. I mean, he received a bachelor and master in science in chemical engineering at the University of Michigan. And then he got a PhD at the same university under supervision of Professor Robert Sips, working on applications of percolation theory. Then in 2001, he moved to Sandia National Laboratories to work on multi-scale simulations. And after that, he moved in 2005 to Iowa State University, also working on computer simulations. And finally, he was appointed lecturer at King's College in 2007, where he is now head of the engineering department. So, and his talk will be about computer simulations of soft nanoparticles. So, I mean, the floor is yours, and thank you very much for coming.
Great. So thanks a lot for inviting me. And thanks to everyone for showing up. I know it's basically holidays now here in Barcelona, so I appreciate you taking time to come along. So just to recognize the people who do all the hard work and the people who give us money and computer time to do our simulations and stuff. So I met Jordi 19 years ago when I first came here and I was a postdoc at Iowa State, and I came to visit Jordi just after my interview at King's College, which then ended up being where I work. And I dug up our paper that we worked together on, and I just found it really fun to see that our simulations, we wouldn't even get past, like, this is just an initial study now, Jordi. They would just reject instantly for 17 nanoseconds of compute time, but things have come a long way since we did all this. And that was the first time I ever studied anything that represented a biological system or biomolecules. And since then, my group now has kind of two different strands. So one is more on kind of functional materials, a lot of the self-assembly things and nanoparticles, which I'm going to talk about today. And then the other is more biophysics, which we do a lot of different peptides, lipids, and now we're getting into mucin, spider silk, and these kind of things. But today I'm just going to talk about some of the work we've done looking at different types of nanoparticles. I'm going to mostly talk about polymeric nanoparticles, but then I'll just show some highlights of some other things we've done at the end. And I'm going to start out by talking about a particular polymer called tetronics. And so tetronics are like an X-shaped polymer that's analogous to the pluronic, which a lot of people are familiar with. And it has a PEO block and a PPO block in each arm. And there are 4 arms. And I did this, one of my former PhD students did all the simulation work, and we did this in collaboration with Cecile Dreiss at King's and Gustavo, who's at Navarro here up in the northeast of Spain. And so tetronics are interesting for various reasons, but one thing that I've always kind of found interesting is that they'll form these pseudo-polyrhodoctanes wherein you have cyclodextrins, and you can thread them onto the various arms, and that'll allow the micelles to break up. With Cecile and Gustavo, we looked at 2 different types of tetronics. So we looked at one that's small, so they only have 4 of each monomer type in the arms, and then the larger one, which has 17 propylene oxide and 15 ethylene oxide monomers in each arm. And with the small one, we looked at the self-assembly, which I'll talk about in a second. But in the larger one, because it's so large with our simulations, and because we were using all-atom molecular dynamics simulations, we just had to pre-assemble the structure and then try to understand the internal and interfacial properties of these nanoparticles. So one thing that Rob figured out when we were looking at these things from a simulation perspective is just that when a lot of people look at the equilibration of nanoparticles, they often look at the radius of gyration, and they find that the radius of gyration, once it's kind of plateaued or reached some kind of constant. They say that the simulations have equilibrated. But what we found is that, so Rob was interested to look at what happens to the internal structure. So what he did is look at the evolution of the placement of the ethylene oxide monomers within the micelles as a function of time. And so even though the radius of gyration equilibrates at around 50 nanoseconds or so in our simulations, we don't see the internal structure of the nanoparticle really equilibrate until 100 nanoseconds. So there's a kind of two different timescales on which things equilibrate within these kind of nanoparticles. There's the overall morphology or shape of the nanoparticle, but then there's the evolution of the internal structure. And so for us, we always, since we're interested in being able to give as detailed a description of what's the internal structure looks like, we try to utilize this as our metric to determine whether our simulations have reached some kind of equilibrium or not. And another thing that we've done some work on, so with these kind of nanoparticles or any kind of soft interface, obviously these are not perfect spheres, so there's a lot of roughness on the surface. And so in order to try to be able to characterize these interfaces as well as possible, we utilize what's called intrinsic surface. And so in this case, an intrinsic surface just means you use some kind of reference from the molecules themselves to define the interface at a particular region of the nanoparticle. And so one of my early PhD students, Dan Allen, did some work where we used one method. This is just of a SDS micelle. And here in his method, we used the sulfur in the SDS head group. And so that's a pretty well-defined atom that we know is probably going to be at the interface of the hydrophobic hydrophilic interface of the micelle. And so with him, we were able to develop some code that allowed us to be able to develop an intrinsic surface for a micelle when you have well-defined kind of anchor points. And you can see the difference that, so basically this is if you just use the center of mass of a nanoparticle and then measure the density or whatever property you want to, but in this case density, as a function of distance from the center of mass. And this is what happens if you account for the intrinsic surface, so you get a bit more detailed perspective. And you can see that if you take just the radial density, you don't get the slight hydrophilic peak that you see at most kind of charged surfaces in the water density, whereas you do when you do intrinsic surface. And this is because you're kind of averaging out in different chemical environments as a function of radius. Whereas here, since you're always measuring from the distance of the interface in the local region, you're getting a more like-for-like comparison. And so the averaging kind of doesn't have the same effect. So anyway, so it turns out we've done a lot of different work in order to try different ways to try to do this, to create these intrinsic surfaces. So we've written some code that we share with the community to do sphere-like nanoparticles, and more recently, we've written some code that allows you to do aspherical nanoparticles. And so that, because this won't work as well for aspherical, but here we've used convex hulls and alpha shapes, and it allows us to be able to do more like rod-like nanoparticles. And previously, we've also done some work to try to do it for soft interfaces like monolayers or bilayers as well. So using these kind of methods, then we can characterize the interface of these large polymeric micelles. And for the most part, we see that the PEO is this purple curve, the PPO is the orange curve, and then the light blue curve is water. And we can see that, of course, we have this water interface. Again, we have this slight hydrophilic peak. But the interesting thing that in all the EO containing kind of nanoparticles we study is that while, of course, EO forms the corona of the nanoparticle, we always see some EO kind of interdigitate into the hydrophobic core of the nanoparticle and take some, and so as a result, some water penetrates into the core as well. And you can see that this kind of peak in the EO kind of aligns with this dip in the PPO density, and so there's some some kind of alignment of that. So what we wanted to do then is try to understand better what the internal structure of these polymers are inside of the nanoparticle. And because of the fact that this is a complex geometry, Rob and I spent a lot of time trying to figure out how we could come up with some geometric method to describe how these different forearms are oriented within the nanoparticle. In the end, Rob came up with a way to try to do this with machine learning. And so we used 8 different distances to characterize the shape of each monomer. So effectively, those 8 distances are from the center of this center part to the edge of the propylene oxide block in each arm, and then to the terminal monomer and the EO block in each arm as well. So 2 distances on each arm, and so that gives us 8. And then what we're able to do is feed that all into a dimensionality. So we take those measurements for every molecule and every time step of our snapshot of our configuration. We feed that into a UMAP, which is just a dimensionality reduction code algorithm. And that gives us then this 2-dimensional representation of our high dimensionality data. And then we use HDB scan, which is just a clustering algorithm. And that allows us to then cluster the conformations of these polymers. And in this case, we find 5 different clusters. And then what this shows is these are just the histograms of the normalized input distances for each cluster. So you can see that this just shows that each of the clusters are unique. So while some of the histograms may look similar, there's never one that's completely the same. So it just goes to show that the clusters we're getting out are informative and not just kind of artifacts of anything. And then we're able to get representative structures of each of the different conformation or clusters. And so we can see some like this one where there's a polypropylene oxide part just clustered together, and then the EO arms are all oriented in different directions, reaching out to the interface. And then this one, where you have one arm reaching out and the rest kind of sitting maybe on the surface, and et cetera, et cetera. And these show the probability of each one happening, or the density or occupation of each one. And then here we can show, because we can label each molecule, then we can identify where they're at, and we can try to see if there's anything that shows us anything interesting in terms of the relation. So here we've just colored all the different polymers by the cluster they sit in. And so we generally can see that like clusters 2 and 3 are close to the core shell interface. 1 and 5, they reside more in the core of the micelle. And then 4 is where you have 3 at the core shell interface, and then one arm kind of reaches across the micelle to the other side of it. So in addition to trying to be able to understand the conformation of these molecules within the micelle, we also wanted to try to understand the dynamics of the EO blocks in the T904. So Rob put together a hidden Markov model, and we identified 3 different states, so contracted, intermediate, and extended. And what we can see is that, so this just shows the distance as a function of the EO monomer in the chain, so starting with the one closest to the polypropylene block, and then the terminal one here is EO15. And so we can see that for the contracted, basically, we see a slight increase in distance, and then the terminal ones come back to the surface, and then the intermediate ones, and then the extended ones, as you would expect, are further and further from the interface of the micelle. We also looked at how many waters bridge from one monomer to the interface. And obviously, these are all averaged over every conformation or structure and time step. And again, we see that for the contracted, there's the terminal part that bends back toward the surface, has short hydrogen bonded bridges, And then the further and further they get, the longer and longer the chain, as you would expect. And we see that basically as you move further from the interface, the more water's hydration you see around the various monomers as well, which again makes sense. So we used all those different techniques to try to get a better understanding of just the dynamics and structure of the molecules within the larger polymer nanoparticle. And then another trick we use is to use graph theory kind of approaches to model self-assembly. And we're not the only, lots of people do this. But in order to do this, all we have to do is just we just use graph theory. We use some cutoff to determine whether things are in contact with one another. And if so, then we can represent the interaction between these two molecules with an edge between the nodes, which represent the different molecules in the system. And then you can easily put together an adjacency matrix where you put 1 if the two nodes are in contact with one another and 0 if they're not. And we use this for the hydrogen bond chains. We use it for measuring self-assembly of molecules when we're looking at self-assembly. And so using that tool, then we wanted to investigate the self-assembly of these smaller tectronics. And when we first did this, we ran a simulation just kind of naively of these polymers. And we had it completely, no net charge, just completely neutral. And we ran the simulations and we see that everything aggregates into one big micelle. We have 30 polymers, they all aggregate into one big micelle. And then we talked to Cecile and Gustavo and they say, something's wrong with your simulations because in experiments we see almost no aggregation. And so this is where kind of having someone who has a different background working with you is good because Rob is a chemist by nature. And so he instantly, well, after some time, thought about pH. And so he asked Cecile and Gustavo, what's the natural pH of the polymer? They went and figured it out that it's 9.7. And so then, And these are different pHs. These are like the curve here you see is, I think blue is acidic, orange is the natural pH, and green is basic, I'm pretty sure. But these are from the light scattering experiments, and you can see then the difference, how pH affects the aggregation size, or the size of the micelles that are formed as a result of changing the pH of the experimental system. And so of course you can do constant pH simulations, but those are very timely and we're a bit basic in our group. So we just decided to just create models of different protonation states of the polymers and use those as an approximation for the effect of pH. And so on the right, we have singly protonated polymers. Here we have doubly protonated polymers. And I love these videos because they look like kind of like humans falling in like skydiving, I think, which is always fun. And so we can see the difference in the aggregation. We can see more aggregation in the singly protonated, while in the doubly protonated, maybe not so much. And so then we can start to try to understand more quantitatively how the aggregation size varies with pH. And you can see, just as we saw in the video there, that for the singly protonated, there's much longer tail in terms of cluster size as opposed to the doubly protonated. And we can measure radius of gyration, radius of hydration, and then this is the eccentricity of a measure of how spherical the micelles are for the two different systems. And we see that as it gets more charged, we see the aggregation number decrease, and we see that the radius of gyration, radius of hydration changes, and both of them stay at about equally aspherical from one another, where 0 would be spherical. And when we compare this to the light scattering experiments, they found that the acidic pH has a radius of hydration around 10 angstroms, which is in quite good agreement with our doubly protonated system. And their natural pH has a radius of hydration of 13 angstroms, which again is in surprisingly good agreement with our singly protonated system. And so we wanted to then try to use our simulations to try to understand what's happening to cause this difference in the aggregation. And so what we found is we looked at the hydrogen bonding between these two nitrogens in the central part, the cross-linking part of this star-shaped polymer, and we saw that we see that there's strong hydrogen bonding between those two groups when we have single protonation. And then when we have double protonation, we see two distances occurring between those two nitrogens. And then for the unprotonated, or the completely neutral one, we see the more extended of the two conformations that we see in the double protonated case. And so then when we see we started to think about what that means in terms of the dihedral, or the conformation of these two nitrogens. And what we found is that in the single protonated case, we see that these nitrogens take a cis conformation. And then in the double protonated, we see that there's a mixture of trans and cis. And then the unprotonated, we see it only take trans. So in the end, this hydrogen bonding results in conformational change of that central core nitrogen group unit, and then that affects the amount of hydrogen bond, or the hydration of the head group or that part, which then results in the decrease or increase of aggregation. So after we studied a bunch of these tetronics, then another PhD student, Raquel, came along, and she was interested in studying PEG and PLGA polymer nanoparticles. And in particular, she was interested in this EEK peptide. Is a peptide that has been patented by my collaborator, Martin Olmsnyder. And so we were interested, and it's an anti-cancer peptide. And so we were trying to understand how these EEK peptides can be encapsulated into a PEG and PLGA polymer nanoparticle. And so for these simulations, we moved from atomistic representation of all the molecules to using the commonly used coarse-grained force field Martini. And we have 200 of these polymers inside of our system, and then 15 of these peptides. And again, we wanted to first try to understand what the internal structure of these nanoparticles are. So in order to do so we measure we use intrinsic surfaces again to measure the densities as a function of distance from the interface. And the blue curve is water, the pale blue curve is the PEG, and then the pink is the lactic acid, and the darker pink is the GEA. And we can see again, these peaks of water and stuff in the middle look really large, but of course these are small volumes, so they're not as massive. The size of the peaks are slightly enhanced as a result of that. But this shows then the fraction of beads that are within the core of the different species. And again, we can see a decent amount of EO inside, and even more than in our atomistic simulations. And we see a little bit of water and an even little less amount of peptide, but we do see some peptide in the core. And otherwise, we see them sitting at the interface. But we see some peptides get into the core, and we can see some water that's sitting next to them. So we wanted to try to understand that a bit better. So the first thing we wanted to do is to understand what's the difference between the interaction of these peptides with water and the polymers when they're at the interface of the core and shell, and then when they're inside the core of the nanoparticle. And so what this just shows the change in the number of waters that are in contact when we take, and if it's greater than 0, then it means there's more contacts with water inside the core than at the interface, and if it's less than 0, then it's more at the interface than at the core. And so we can see that, of course, at the interface, there's a lot more hydration of these peptides, which is noted by all the significant number of negative numbers here. But there are certain peptides that are slightly more hydrated, or residues which are slightly more hydrated within the core than outside. And then if we look at the polymer EEK contacts, again, we see that inside the core, of course, there's a lot more contact with the polymer than there is at the interface, because now it's completely surrounded by polymer. But again, there's some residues which are more preferred interacting with at the interface than when inside the core of the nanoparticle. So then again, we use our trick to characterize the conformation of these polymers, the same kind of ICMAB trick. In this case, we just have two distances. We just have the distance between the two the terminal EO blocks or monomers, and then the distance between the two terminal PLGA monomers. And we again use this with HTV scan and ICMAB and to come up with our clusters. And these are the histograms, again, just to show that they're unique clusters. And then here, again, these are just kind of representative conformations of each cluster. But the cool thing that we were able to do then is to then try to look to see where these clusters are within nanoparticle again. But even more interesting is now we can try to understand which ones are in contact with the peptides, whether it be at the interface or within the core of the nanoparticle. And so while we see at the interface, there's no real preference, it's kind of more or less random which ones the peptides are interacting with. Within the core, we see that there is one preference, or there's a fairly significant preference for, although the error bars are, of course, relatively large because we only have one peptide inside, but nonetheless, from what we can see, that there is a preference for the type of, or the conformation that the polymers take when interacting with the peptides inside. And what we can see here is, so these are this is the PEG part of the polymer, this is the PLGA part, this is the peptide that's interacting with the hydrophobic and a bit of the PEG, and then this is the water that's kind of surrounding it. So we are able to see that there is some kind of local environment where these peptides seem to bind. The only question we can't really answer is whether if it's a chicken or egg, because of course the water is there too. So that is something that we weren't quite able to assess, but nonetheless, we're able to provide a bit more insight. And I think this demonstrates that using these kind of tools, we can now look more carefully for what kind of local environments might be found. And maybe this is another kind of design criteria that one might have to consider while trying to understand how to optimize encapsulation within these kind of the formulations. Then after that, we wanted to start to try to understand, of course, how these things interact with membranes. And then in Raquel's case, she was interested in trying to understand whether they interact any differently with cancerous membranes or healthy membranes. So this is just like a red blood cell model. And then this is a cancerous model, which we have from lipidomics from some cancer cells, which have been studied within Martin's lab and his collaborators. And so the first thing we wanted to do is just understand how the radius of gyration of these micelles change as they get closer and closer to the interface of the lipid bilayer. And so we see that in the case of the cancer cells, when interacting with cancer cells, there's a general kind of contraction of the core of the micelle, whereas the overall size of the micelle increases, which is slightly different than what we see when we look at the healthy. It's interacting with the healthy membrane, which is when we see with the healthy membrane that the overall size increases, but then that the core of the micelle seems to keep more or less the same size. And of course, we see it kind of cause indentation within the membrane as we start to see it interact with it. So we wanted to try to understand a bit better what's happening here. And that increase in the size of the whole micelle is a result of the spreading out of the EO arms on the lipid membrane. So as it comes into contact with the membrane, we see the EO arms more or less uniformly spread out onto the interface of the lipid bilayer. Some of them we see kind of reach across the membrane into the other leaflet and start to try to sit at that interface to maximize the EO contact with water. But the interesting thing that we've seen so far in this work is that, so the nanoparticles go to both membranes. So it doesn't seem that in that sense it's particularly selective, but it does have different, it does seem to interact with the membranes in a slightly different way. And in the healthy case, we see that it doesn't, while it does thin the membrane a bit, it doesn't thin it anything to the same level as when it interacts with a cancer cell. And we seem to see that thinning of the membrane seems to increase some and gets more localized as the simulation progresses. We also wanted to just see if we could see, now that we have these peptides in the nanoparticle, do the peptides move once, or how do they go from the nanoparticle to the membranes? And what we found is that in this case, so this shows like the maroon color, brown color is the peptide, our particular peptides in the nanoparticle, and then the green color here is the membrane center of mass, and the green color is the nanoparticle center of mass. And so here we see that these peptides seemingly, in the healthy case, more or less move with the nanoparticle itself. So if the nanoparticle is rotating around on the interface at all or moving in relation to the interface, then the peptides move with it. So they don't seem to necessarily diffuse independently of the nanoparticle. Whereas in the cancer case, we don't see the same coupling between the nanoparticle, which again is this green color, and the peptides, which are these brown color. So in that case, it seems that the membrane, the cancerous membrane, which of course is more heavily charged negatively and the peptides are positively charged, then the peptides seem to be able to diffuse along the surface of the membrane to move closer to the interface of the membrane itself. So this part with the membranes is stuff that we still are in the process of trying to wrap up, but so far it seems like some kind of interesting results and gives us a bit of a better idea of at least how these nanoparticles work in contact with reasonably representative membranes. Just to give a flavor for some of the other type of nanoparticles we've looked at in the group. So we've looked at lipid nanoparticles. These aren't the cool ones that became super excited when with the coronavirus vaccines, et cetera, but these are made of triglycerides. And so we've looked at solid lipid nanoparticles. And in a second, I'll show some pictures of liquid ones as well. But so in this case, we have this tripalmitin, which forms the core, and then a surfactant, brige, which is used to stabilize the nanoparticle. And one thing that I wasn't anticipating when we did this is that the triglycerides form facets within the solid case. And then there's these kind of ravines or valleys of hydrophobic tails where the surfactants kind of sit amongst and kind of protect the hydrophobic tails from its interaction. And this just shows how one single molecule, kind of how its tails of the bridge kind of interdigitate with some of the hydrophobic tails of the triglycerides. We've also looked at liquid lipid nanoparticles, where the only difference is just that the tails have the double bond here, so those kinks make them liquid instead of solid. And again, we use the same bridge surfactant. And here we use a different approach, but the same general idea of dimensionality reduction and clustering. And we're able to find different clusters. These are all the same. So if you look through the literature, people have been classifying these molecules using just geometric measures before, and they've found all these same kind of conformations. So the interesting thing here was that we were able to use machine learning to classify these molecules, and the clusters that we found basically reproduced the conformations which people have been identifying using different or more geometric methods for a long time. And so it kind of gives some confirmation that the machine learning is doing something useful. We've also done some work on surfactants, and we've been studying Triton micelles for a bit now. And the interesting thing that we found, so we've looked at now 5 different drugs loading into Triton X100, and then we also did 114. But so in Triton X100 and 114, we studied ibuprofen and then endomethacin. And ibuprofen encapsulates into the core of the micelle and causes the micelle to become elongated. But with indomethacin, the interesting thing we found is that in both cases of Triton X100 and Triton X114, which isn't surprising because they're very close, but is that after it aggregates, then the interaction, because the drugs interact with each other so strongly, then it actually causes the micelles to break into one large micelle, break into 2 smaller ones. Yeah, and I think it's just because the surface area coverage of the surfactants, when all these things aggregate together due to the strong interaction of indomethacin, isn't enough to kind of shield the hydrophobic core anymore, and so it needs to break into 2 smaller ones. But then when we've looked at, more recently, we've looked at these 3 drugs, which the log p-values of indomethacin and ibuprofen are just a little bit lower than felidipine. In this case, we don't see them interact, and they don't go into the core of the micelle at all. Obviously, felidipine is quite hydrophobic, so it likes to encapsulate, but it all sits at the interface of the hydrophobic core and hydrophilic head group, because Triton again has these ethylene oxide head groups. And then we see more or less a direct relationship between encapsulation and log P in this case. So anyway, so it just again reiterates that there's some, that the chemistry of the drug plays a pretty important role in the final encapsulation of these systems. And then we've looked at things like the lipid digestion. So we didn't actually model the digestion of lipids. We just took the kind of more traditional route of looking at different percentages of the degree of the digestion of a short chain lipid like this into its 2 byproducts, and then we studied the different micelles that are formed at different. So like this is all product or all reactant, 25% product, 50% product, 75%, and then all product. And we did this initially in combination with some really cool neutron scattering, time-evolved neutron scattering, where they're actually watching the systems evolve as the reaction occurs. And and we saw a really good agreement between our MD and the neutrons in terms of size and shape and stability of the micelles that result. And then more recently, because as we were doing this, Demi, who was doing all the hard work, she realized, or she came across the fact that, these two products are actually lipid, they're in permeability enhancers. So we thought we should look to see how they interact with membranes, because it's another, in a way, these could be kind of clever formulations for drug delivery. And so we did some work trying to understand how the different components of these micelles at different stages of digestion interact and affect the permeability of membranes. And then more recently, we've gotten into peptide self-assembly and the nanoparticles they form. And so we did some work with Max Ryadnov, who's at National Physics Lab in the UK. And this is like a triskelion of lactoferrin, which is a naturally occurring antimicrobial peptide. And so we tried to model the nanoparticles that he forms experimentally of this antimicrobial peptide. And the first thing that came out of our simulations was that in order for these nanoparticles to be stable, you have to have a bilayer. So you can't just have a monolayer of these peptides that form a single football like this. Otherwise, it just falls apart because of the interaction of water with the hydrophobic residues that don't allow it to stabilize. So you actually have to have at least a bilayer, and then it could be multiple bilayers in order to form this. So we had to write some code that allows us to pre-assemble these things into two footballs like that. And then we looked at them this is again Martini, so coarse grain simulations, where we look at this nanoparticle then coming to a bacterial membrane. Because unfortunately we don't get to see it dissociate. I think it's because Martini is too sticky and doesn't allow the peptides to fall apart within the timescales we have. So in order to try to understand how these interact with the membrane, we just took a platelet of the bilayer atomistically and we put it in touch with another model of bacterial membrane. And we can start to see the pore formation, so we can get some idea of how these antimicrobial their mechanism of action against the bacterial membrane. And since then, we've done a bit more work in modeling a similar type of pseudocapsid. And then thanks to the power of AlphaFold now, you can generate all kinds of interesting self-assembled structures of peptides. And so Javi did some work where he modeled this beta helix kind of structure and the fibers that it forms. And we're able to get some interesting information which complements the experimental work done in Max's group and stuff. So hopefully I've shown you that molecular dynamics can be useful for trying to understand the structural properties of these different self-assembled structures. And through my time of working on this, it's been great to work with people doing all kinds of different experimental scattering techniques, neutrons, light scattering. I feel like simulations are a perfect complement to these kind of methods. We've created various types of analysis methods, and others have created other ones too, which kind of allow us to get more insight out of these type of simulations. And I think now, obviously, a lot of people have studied individual systems, but now with the kind of steps that have been made in automation and experimental chemistry labs and stuff, it's a perfect time for people to start thinking about how we can combine high throughput simulations and some machine learning with the automation workflow in chemical labs, and probably do some real true rational design moving forward. So anyway, I hope that was useful and interesting. And if there's any questions, I'd be happy to take them. Thanks.
So thank you very much, Chris, for this talk. And now we have some time for questions, both from the people here and from the people online from Zoom. So let me start with the people here. If anybody has any question, comment.
Thank you.
Yeah.
It's very interesting for the talk. Thank you a lot. I was wondering about the simulations that you show with the interaction of with the membranes. So, for example, the cell membrane versus the healthy. Can you specify a little bit more about the differences in the membrane, and if you have seen that the differences are related to specific membrane components?
Yeah. So I think the biggest difference is that I can't remember the specific lipids. I can get that for you, but the biggest difference is the cancer cells are more negatively charged. So I think they have more PS, phosphoserine head group lipids in the external and the leaflets than the healthy cell does, because obviously in healthy cells they don't expose negative lipids to the environment. So yeah, I think that's the main difference. So it's maybe not surprising that the peptides, which are positively charged, then kind of diffuse through electrostatic interactions more to those surfaces. But yeah.
Thank you. Yeah, I was wondering if there were maybe also some membrane proteins, but it was a lipid.
Yeah, in this case, we don't mess around with, we try to, we keep it physics-like and keep it simple. Yeah.
Thank you.
You're welcome.
More questions? Comments? Let me see whether we have any comment from the people in Zoom. If I'm able to see that, let me see. It has to be the chat here. Oops, the mouse here. Okay. So let me see. We have 6 people online, but okay, apparently, I mean, I suppose I'm doing that correctly. We don't have any question from the chat. So I will give some time for the people from the chat to make questions, and I will do also questions. I'm curious about the machine learning methods that you use to classify the different polymer configurations. And these are methods based on, I mean, they are classification methods, neural networks, or what are they based on?
So they're just unsupervised learning techniques. So basically, for every molecule, we just pass a vector. You can use any inputs that you want. So we've done this for peptides, like for antimicrobial peptides too, that don't have any well-defined structure, or we're interested to see how their structure evolves. So, and usually what we found is that, so we always take intermolecular distances. We've also done it for lipids to try to understand how cholesterol interacts with sphingomyelin. And there we used intermolecular, but for the most part, we use intramolecular distances. And the trick that what we found is that the fewer distances you use, the better clustering you generally get. But with like peptides and stuff where we don't really know, we don't have a well-defined, with polymers, it's a bit easier because you can kind of pick places because you know where the hydrophobic and hydrophilic blocks are. With peptides, we found it was more difficult, so we just used all the distances between the C alphas. And in that case, then we just filter out all the distances that have low variance at the end. So like if there are parts of the peptide that with relation to one another aren't really moving much, we just throw those out before we send them to machine learning, because what we're interested in are the high variance things, because that's going to be what governs your conformational changes. And then you just create a vector, and then the important thing is that once you've created these vectors for each conformation at each time step, then you normalize the distances in the column, like for every distance over all the time and stuff, you have to normalize it so it's between 0 and 1 instead of using the raw data, because again, we found that it clusters better if it's just clustering numbers, like a smaller range of numbers. And then you just feed it to this UMAP, which is a one-line command in Python. And then it'll give you back its reduction of the dimensionality to two dimensions, and then you feed it to HDB scan. And I think there's 2 hyperparameters in UMAP that you can kind of play around with, and really 1 in HDB scan, and then the rest is So, and then it outputs the clustering, and then you can kind of play around with it to see with these hyperparameters to get kind of something that looks reasonable. And then we try to, like I tried to highlight here, we try to use different, so for the polymers, we always create these histograms to show that, to confirm to us that we're looking at unique clusters. With peptides, because we have so much more data, trying to do histograms is a bit trickier. So what we've done there is just use secondary structure, calculations to show that the average secondary structure of the peptides are unique to one another. So again, we feel fairly confident that the clusters we're identifying are unique, or different from one another. So, because obviously you can create as many clusters as you want, but if half of them are the same structure, it's not really enlightening. So yeah.
I'm also curious about the bacterial simulations.
Yeah.
The membranes that you consider were from bacteria gram-positive, gram-negative.
Yeah, so in these simulations, I think we've taken so a lot of our collaborators, when they do it in the experimental lab, biophysical things, they just do like purely PG lipids for gram-negative, I think, and then a mixture of PE, PG, like 70-30 for gram-positive. But more recently, in other work, we've been now doing more realistic model membranes where you also have the lipopolysaccharides and the gram-negative outer membrane. Obviously, when you start to mess around with those, then having the diffusion of things through them is very slow, right? And so what we've been, but I think, at least the way I understand it and speaking to the experimentalists, is that it's not very well understood what the actual amount of LPS is in the outer membrane sometimes, because it's hard to characterize this. So usually what we do is we just simulate membranes with varying amounts of the LPS, and then try to understand how things diffuse through the less dense ones. But yeah, so we've done both, but in this case, it's just been the very generic models. Yeah.
Okay.
Yeah, just one naive question about the time that would take to simulate another nanoparticle, like the one that you showed of PLGA.
Yeah.
How long would it take to create another nanoparticle model?
So what I usually say is that I think if we do it at coarse grain, that same resolution where we're not modeling every atom. So coarse grain just means we group atoms together, and it allows us to simulate longer or larger systems. But it would take about, I think, 2 to 3 weeks to do the simulations and another week to do the analysis, basically. So around a month is usually what we kind of say. But yeah, we have all the tricks and stuff to do it now. It's just a matter of finding someone to do it, but it's not prohibitive. And yeah, I mean, just like Jordi, we're pretty lucky that in London that we have access to a lot of computers. So compute time's not really an issue for us.
And just another quick question about the simulations that you've showed with the cells, either cancer or healthy cells.
Yeah.
Are you always considering just one nanoparticle with the membrane? And do you think that maybe having other particles would change that interaction?
Yeah, so I'm sure. So the answer to the first question is, yeah, we've only looked at one just because, again, the size and scale. So with these simulations, we can kind of do, with coarse grain, we can do maybe tens of micron by 10 of micron kind of swath. So, and the nanoparticle, I think in this case was, I don't know, 30 or 40, like 3 or maybe 5 to 10 microns in size. But yeah, so we only look at one. I think definitely that it would have some effect in the sense that they could aggregate potentially, or they could interact with one another at the membrane surface. But in that case of that one that I show you there, our initial simulation, the simulation box and like from the cross section wasn't large enough. So those EO arms were actually kind of interacting with each other in a sense already. So, but it would be an interesting thing to do, to try to look at more, but we just haven't figured. I mean, I think Jordi's got some interesting methods I read about in the thesis that I'm here to examine that might be useful for some of that, but yeah. Hey.
Thank you. Great.
Good to see you, Andres.
It's always nice for me to see simulations going into the real world, and now more and more. So I wanted to ask about the last part, the next steps into bringing together in the automation, simulations, experiments, now machine learning. In your case, where do you see it should head, the molecular simulation part, to go into this? I'm now also working for material science. So we are at the Catalan Institute of Nanotechnology. And the idea is this, not only using machine learning because we have the data, but bringing the simulations also into this workflow of design of materials or peptides or.
Yeah. So I think that, so my whole career, I've been trying to do the most difficult, the most realistic models and stuff, which I think is useful. But I think to do this now, well, first of all, I think that things like Martini are advancing so quickly. They're about to release like a model of pep proteins that can fold in Martini. So this is just going to allow us to do more realistic simulations faster, right? But I think there's definitely room to think about what's the simplest model you can put together for simulation to get the information you want, and then using high throughput methods to basically spawn out as many simulations as you possibly can to collect the data you want, and then do some filtering, right? Because now with the automation, the experimental side, they're basically using robots to do the same thing, right? They're trying to produce, at least the ones that I talked to at King's, the people are trying to do experiments in droplets and all this kind of stuff, where they have robots that drop all the materials in for the chemical reactions or produce polymers or whatever they're trying to do. And so to me, it seems like you could do simulations with coarse grain simulations as large number as you possibly can, all the analysis now is scripted, so we can just do automated analysis. You look at someone like this guy at MIT, what's his name? Anyway, there's this group at MIT who I almost did a postdoc with, and I forgot his name now, but he basically has written a machine learned algorithm that creates hypotheses, sets up the simulations, runs the simulations, analyzes the simulations, and supposedly writes the papers, which I'm not sure how journals feel about that. But anyway, I think that this kind of stuff is it's here now, and we just have to figure out, just like we used to have to talk to people to understand. I had to learn what a peptide meant, like what they meant when they said radius of hydration instead of radius of gyration or something. Now we just have to sit down and figure out how can we take inputs from simulations to be useful into experiment. And then I think in 5 or 10 years, it'll be just people run everything together, and it's just a self-driven lab basically in all avenues. But I think moving back to simple things slightly to get the information we want, and trying to do it as a screening process, and then going more and more detailed when you want to get more information. But I think with the speed that experiments are moving toward, in a way that atomistic stuff is just going to be a more way of understanding what's happening, as opposed to, I think that atomistic simulations, unless we figure out how to do them faster, are probably too slow to keep up with the way experiments are going to go, I think. That's my feeling.
And now I see that we have questions from the people online. So let me see if I'm able to see that. Okay, it's here. Okay, it was not this, but it's this window. Okay, so it was my mistake. Okay, so here you have two questions, one long and the other one short.
Yeah. So the first question is about whether we've done any backmapping. So backmapping means you can take the Martini, which is kind of coarse-grained representation, and then transform it back into atomistic simulations. And in fact, we have done a bit of that. So we've just done some of that actually in collaboration with Bepi Battaglia, who's in Barcelona here. So we're starting to work on some of that. But not to address the exact question of the stickiness of Martini, but we have started to play around with this in a way of using Martini to do the self-assembly and then use atomistic to gain more insight. And the second question is about whether we've applied this machine learning approach to both coarse grain and atomistic, and in fact we have. The first simulation, the tectonic simulations I showed the outputs were for atomistic, and then in the PEG PLGA, that was coarse grain. So that's an example of how we've applied both, and they work in both cases. Because basically you're using the same same metrics. And in terms of which kind of cancer cells, Jordi, those are breast cancer, I'm pretty sure we were trying to model. So again, I'm sorry I don't have the lipids, but I can figure it out. If people want to email me, I can tell you the composition, I think, or at least I can make sure some of it Martin's kind of protective of because he's gotten from lipidomics. But anyway, I think these are probably from a paper, so I can figure it out.
Okay, so I think that we can thank Chris again, and if you have more comments, we can talk with him. He will be around during this morning.
Great. Thank you. Thanks a lot.